My beautiful people, so as we are preparing ourselves for final exams, I want us to look at random questions in biology, and we are going to use different past papers. So we have the first question under the cell structure. Figure 1.1 shows an animal cell as seen under an electron microscope. So an electron microscope is a type of microscope that at least gives us a detailed and more clear picture. So we have been given an animal cell with an identified parts labeled I, F, G, H, and also we have an identified one, which is the cytoplasm. So the first question is, we are taught to identify. So to identify simply means to name the given structure. So we are taught to identify the labeled parts F and G. Now, before we start answering our questions, let's ask ourselves, or let's define what a cell is. So a cell is seen as a basic unit of life, which simply means this is the core of life. That's where life is. Okay, so part F is this structure. So we have this structure with these ribosomes on the surface. So whenever you see a structure like this one with the ribosomes on the structure, is called a rough endoplasmic reticulum. So presence of ribosomes gives this the name of rough endoplasmic reticulum. Then absence. So if this structure has no ribosomes, then would have named it as smooth. So the difference between smooth and rough is that smooth does not contain the ribosomes, while rough contains the ribosomes on the surface. Then what of part G? So part G is this line pointing this circular structure, and this one is called a nucleus. So we've got a nucleus here, and a nucleus controls whatever activity, or we can say controls whatever reaction that occurs within a cell. Then inside the nucleus is where you can find structures like we have chromosomes. Then chromosomes contains the gene. The genes contains the protein, the DNA, the RNA, stuff like that. Then we move on to question B. So question B, we are told to explain the functions of the parts labeled H and I. So H is this sausage-like structure, and this one is called a mitochondria. So we have a mitochondria. So the mitochondria is sometimes known as a powerhouse, and there is a reason why it is called a powerhouse. It's called a powerhouse because this is where energy is released. What is the importance of energy? As we know in science, energy is the ability to do work, and we are saying the cells have works, they have specified work. The cells, each cell has a specialized work it does for life to be possible. So for work to be done, it requires energy, and that energy used by the cell is generated by the mitochondria. So that is our H. So we can say it is also known as powerhouse. So it is a site for respiration. So if we use respiration, we know that we are looking at the breaking down of glucose to release energy. So it's same as it is a site for releasing energy from the food that we eat. Then what of I? So I is the outer structure, and this is called the cell membrane. So the membrane is a plastic-like structure that covers a certain structure. So now, in our case, when we look at this cell, whether being a plant cell or an animal cell, the function of cell membrane for an animal cell is the same as the function of the cell membrane for a plant cell. So it acts as like a security guard. What do we mean by that? We say it is a selective permeable membrane, which means it selects, it allows substances that are useful to enter and substances that have no use to exit. That's why we say it allows some substances to enter and leaves the cell. Then we move on to question number 2. Suggest 2 cell parts which would be present in figure 1.1 if it was a plant cell. So this question is indirectly telling us that it's like we are looking at the difference between a plant cell and an animal cell. So if the given cell was to be a plant cell, what 2 structures would this cell possess? So structures like cell, then we may have cellulose, we also may have chloroplast. And also, it's also important you understand the structure and its specialized function, or let's say functions. Right, so we move on to the second question, also under cells. So we have 3 cells labeled P, Q, and R. So as the question reads, figure 1.0 shows some specialized cells P, Q, and R. So based on the structure, we are able, or we should be able to know the type of the cell and whether it belongs to animals or plants. So we've got question A, name each of the cells above. So the first one is P, and so what will help us to identify P is we've got this elongated outgrowth known as a root hair. So if this is a root hair, then the cell is called a root hair cell. So that is for P. Then Q. Q, we've got a biconcave kind of a shape and it is irregular in shape. So whenever you see a structure like this one, just remember that you're dealing with a red blood cell. So a red blood cell is one of the components found in blood, or one of the components that forms the blood. Then we have R. So R is, so there should be here, we can see we have a lot of chloroplast and cells with these structures are called the palisade. So we have a palisade cell. Then we look at question. So B1, state the function of P and Q. So P, we say it is a root hair cell. So for root hair cells, these are cells that are found on the roots, and we know the function of the roots are to anchor a plant, then also for absorption of some materials from the soil into the plant system. So function of cell P, we can say to absorb. So to absorb the mineral salts. So the mineral salts and water. from the soil into the roots. Then when these materials are absorbed, they need to be transported by the xylem to the leaves to do what? So that photosynthesis may take place. And we know the importance of photosynthesis is so that food is being manufactured. Then function of cell Q. So cell Q is a red blood cell. Number one, characteristics of adaptation of red blood cells is that they do not contain the nucleus, and for that a large room or space is created so that my hemoglobin may occupy that space. So now function of cell Q, we can say to transport oxygen. So to transport oxygen and a small amount of carbon dioxide. Then Roman number 3, explain the adaptations for cells P and Q to their function. So adaptations are things that makes the cell to carry its given process. So like for cell P, which is we say it is a root hair cell. So what is its adaptation? So we can say presence of outgrowth called root hair. Then adaptation of cell Q, absence. So absence of nucleus. Remember, so that a more room is created. Then we look at question C. Where in the leaf is cell R located? So cell R, we say it is a palisade. So the layers of a leaf is where this cell is located. We can say palisade, mesophyll layer. Okay, so let's move on to question. This is question number 3. So question number 3 is coming from the topic of enzymes associated with characteristics of enzymes. So we have figure 2.0 shows a graph showing the effect of temperature on an enzyme catalyzed reaction. So here we are looking at the effect of temperature. What happens when the temperature increases or decreases in respect with enzymes activity. So we have in the y-axis rate of reaction, x-axis we have temperature in degrees Celsius. The first question is, what is an enzyme? So an enzyme is not a catalyst. It's not a catalyst, but it is a substance that is produced by a living thing which acts as a catalyst. So it only acts as a catalyst, but in actual sense it's not. So we'll say is a substance produced by a living organism. You can give the definition that you know. So by a living organism which act as a catalyst. We move on to Roman number 2. What is the optimum temperature of the enzyme reaction shown in the graph in figure 2? So the optimum temperature, the word optimum simply means like the best, the point where something is operating at its best level. So how do you find the optimum? Let's say optimum of something. So the optimum is the highest point of our graph. So the highest point of our graph is here. This is the highest because the graph did not exceed this point. So now we need to look at these dotted lines. Then the point where these dotted lines cuts the x-axis represents the optimum temperature. So here the answer may not be the same, depends on how good your site is or your estimation. So from my estimation here, I can say 36 degrees Celsius. So if you say 36.5, 36.7, or maybe 37 degrees Celsius, I'm sure we are all okay because here there is no actual demarcation. Then B1. At what temperature in figure 2.0 is the enzyme completely denatured? So denature simply means the changing of the original shape of an enzyme. So when an enzyme changes its original shape, it can no longer fit onto the substrate. What do we mean by that? Let's say this is my enzyme, and as this is its original shape. So the substrate must have a shape in such a way that these two can fit on the active site. Now, if this enzyme is overheated, it can change its shape. So let's say if this active site becomes round like a structure, so we can see that now this substrate can no longer fit here. And since it can no longer fit there, then we can say the enzyme has denatured. And since it has denatured, the reaction now will stop to take place. So that's what it means by denaturing. So now how do we use a graph or graphs to identify where enzymes have denatured? So you need to look at where the curve, this line, is touching the x-axis. So this is the point where the line is touching the x-axis. So at this point, I can just say this is the, I can say 57. So 57 degrees Celsius. So at this point, the enzymes were completely denatured. Give a reason for your answer in A above. So we can say because the reaction came to a stop. Then we have question number last for this question. Explain why it is important to measure the body temperature when a person is sick. So is it just a traditional thing, or why are doctors or nurses measures the temperature when someone is sick? So the reason why they take note of the temperature, so they just want to know if the temperature of the body at that particular time is favoring the activity of the enzymes. So they need to know if the temperature is high, they have to find ways of bringing back the temperature to normal so that the enzymes may not be denatured. Or if the temperature is too low, they need to find ways of raising the temperature so that the activity of the enzymes can be accurate. So to summarize what we are saying, the reason why they measure the body temperature so that they see if the activity of the enzyme is still okay, or to see if the temperature is still in a range where enzymes can operate effectively. So we can say to see if the temperature is favorable for enzymes activity. So it's very important because we know that enzymes, if you look at the characteristics of enzymes, we say that enzymes are proteins. So since they are proteins, the same enzymes, they are found in a cell. And a cell, we say a cell is a region where activities that supports life occurs. So when enzymes stops to work, then the reaction will stop to work, then the cell will die, and the entire organism will die. So that's the sequence. So it's all about the linking. So enzymes are connected with the reactions. The reactions takes place in a cell. Then a cell or cells are connected with the life. So if enzymes dies, reactions stops. When reactions stops, then the life of a cell stops. When a cell dies, then the whole human being dies. Proposed ways the body temperature of a sick person can be brought down to normal. So here we just have to look at the ways that you use as you live your daily life when someone has a high temperature. Like for example, drinking a lot of water helps. Then also wearing clothes that are loose to your body, that are not tight or that are not heavy. So wearing light clothes helps also. Okay, so we move on to question number 4 under enzymes. Figure 2 shows the stages involved in an enzyme catalyzed reaction inside cells of a living body. So let's start the diagram carefully. So there is this structure I. So on stage 1, structure I is the same as this structure. So nothing happened to structure I. Now what of structure G? So here we have structure G, then here we have structure H. But in stage 3, structure G connected with structure H. So they joined together to form a long chain. So we have two substances that are separately, then after certain stages, then they come together, form something that is bigger in size. So from A, question, from stage 1, in figure 2, identify the enzyme. So how do you identify enzyme? So your focus should not only be in stage 1. So you need to look at the first stage and the final stage to help you identify the enzyme. So from stage 1, the substance that didn't change its shape in stage 1 up to stage 3 is the enzyme. So if you look at this I and this Y, never changed. So therefore, we can say enzyme is I, because we say they act as biological catalysts and remains unchanged. That's how we define enzymes. Then substrate can join to form a larger molecule, or we can have a larger molecule breaks to form smaller molecules. So when you have a larger thing breaks into smaller pieces, then those are substrate. Or you have smaller pieces, they join together to form a larger piece. Those are substrate. So the substrate, we can say G and H because they joined in stage 3. Now, which stage shows the enzyme substrate complex? So that's our B1. So enzyme substrate complex, it means the stage where the enzymes and the substrate have combined, they've joined at that point. So the reaction now is taking place. So the stage showing the bonding of these three is stage 2, as we can see. So say stage 2. Now label the active site of the enzyme on figure 2. So the active site is a position on enzymes where the substrate fits the enzyme. So here we can see we have two active sites, because on these two regions is where these two substrates will fit. Then we've got C1. What type of metabolic reaction is catalyzed by the enzyme in figure 2? So metabolic reactions are, we have anabolic reaction, and catabolic reaction. Then the summation of the two gives us metabolic reaction. Now, anabolic reaction talks of building. So here we are building up, we are getting smaller pieces. We combine the smaller pieces to have a larger one. Then catabolic is the opposite, where you have a larger piece and it breaks into smaller pieces. So from what we have seen, we have smaller pieces, then they joined together to give us a larger piece. So this is anabolic type of reaction. So anabolic reaction. Name a region in the human body where such a metabolic reaction in C1 above can occur. So metabolic reaction can occur in organs like muscles. So, okay, let's not say muscles. Let's say a liver. We are looking at organs, so a liver. Now, set the general term used to refer to enzymes that work inside. So there are enzymes that works outside the cell and those that works inside. But our interest is of enzymes that works inside of the cell. And these are known as intra. So inside is intracellular enzymes. Then outside is extracellular enzymes. Describe to industrial application of enzymes. So application are now the uses of enzymes. So for baking, we can use enzyme for baking and for brewing. So we have another question under enzymes. So I also want you to see how the questions under the topic of enzymes are set. So figure 2.0 shows a protein molecule undergoing digestion in the alimentary canal of a human body. So we have the protein molecule structure. So whenever you have a protein, it means this molecule is long. Now, for absorption to take place, we need this molecule to break into smaller pieces. That's where now the digestion process comes in, because we define digestion as the breaking down of larger molecules into smaller ones for easy absorption. So this long molecule of protein underwent reaction K, and it first breaks into two. So you can see we have 2, 2, 2. And there is a reason, and you also need to understand whenever you see a 2, 2. Because 2 simply means here we have what we call a die. So die simply 2. If there was a 3, 3, then we would have said tri. Then here we can say we have 1, 1. So 1, 1 means mono. So that's what it means. Now, let's look at reaction M. So reaction M breaks the di kind of products L into mono kind of products N. So when you look at proteins, so proteins undergoes digestion and gives us the end products known as the amino acids. So the amino acids are the smallest molecules that can easily be absorbed. So here, at least we have an idea. Since these are proteins, therefore, the last part here are the amino acids, because we are looking at the end products. Now, A1, we are taught to identify the product labeled L. So, when a protein breaks into two of these molecules, what is it called? So here, when a protein breaks, it is going to give us what we call peptide. So here we are going to have peptides. Now, since there is a 2, we can say dipeptide. So before amino acids, we have peptides. Since there is a 2, you add dipeptides. So that's where the di is coming from. It means 2. Then suggest a region in the alimentary canal where reaction K takes place. So reaction K is dealing with the digestion of the breaking. Reaction K is the first part of the digestion of proteins. So this one occurs in the stomach, because we know that the digestion of proteins starts in the stomach. Then reaction M is the completion, and the completion of digestion occurs in the small intestines. Now, since K is the starting of this digestion, therefore we'll say stomach. Then we have B. Reaction M completes the digestion of proteins. Which enzyme catalyzes reaction M? So, if these are peptides, then the enzyme should be peptidase. Like we have carbohydrates, we have lipids, lipase. So that's a pattern. Then what is the pH at which reaction M would be catalyzed? By the enzyme identified in B1 above. So the pH, so since reaction M is a completion, the pH here we can say slightly basic. Then suggest where the enzyme named in B1 above is produced. So suggest where the enzyme, so peptides may be produced by pancreas. Remember, pancreas produces the pancreatic juice that contains enzymes that completes the digestion. Identify the products labeled N. So N, these are now the end products, and the end products are the final products that we are getting, which are the amino acids. Now for question number 3, describe 3 adaptations which makes the ileum suitable for digestion. Suitable for digestion, so this should be suitable for absorption. There should be an error. So presence of elongated outgrowth, presence of elongated outgrowth, then presence of lacture. So the lacture is a vessel that absorbs the end products of fats. Then also we can say supplied with numerous blood vessels. Okay, to take place. So we move on to a question under the life cycle in relation with diseases. So figure 3.0 shows the life cycle of a mosquito. So a mosquito is an insect. So sometimes you may have maybe 2 or 3 topics are combined together and they make some questions. So you may have, maybe at times they may give you a skeletal system. So you need to know the skeletal or skeleton found in insects, then also the life cycle, then the type of diseases that can be caused by that insect that you are looking at. So we have stage 1, eggs. So we have eggs here. Stage 2, larva in water. Stage 3, pupa in water. Stage 4, the adult. Then for A1, identify 2 stages in figure 3.0 which are most suitable for eradicating. So to eradicate simply means to get rid of. So to get rid of malaria. So the best one here is, we can't kill the eggs because we know that the eggs have the eggshells. So it's hard to kill the eggs. Then if you look at the larva, so the larva is when the organism is still in its shell. So even if we spray the stagnant water with insecticide, it is unable to feed from that. But for stage 3, it has started feeding, so we can just spray in that stagnant water. So first one, stage 3. Then also the other one is stage 4. Because when we get rid of adult, then we cannot have eggs. We move on for Roman number 2. For each stage identified in A1 above, state how malaria may be eradicated. So stage 3, we can say by spraying. By spraying insecticide in stagnant water. Then is stage 4, adult, we can say by using treated mosquito net. So there are so many ways. Some may use mosquito repellent. So whatever method that you think is okay for that stage, that answer is still okay. Roman number 3, suggest ways in which malaria can be controlled. So here we are looking at controlling of malaria in the community. So the same answers that we have put here can still be the answers for this question, but let's just state another one. So here, since we are dealing with community, we can say by keeping the surrounding clean. Okay, then number 2, okay, we can just say by using treated mosquito net. Then question B, what term is given to the mosquito based on its role in transmission? So mosquito does not cause malaria. It only transmits the pathogen or a microorganism that causes malaria. So therefore, it acts as a transport. So whenever you look at animals that act as a transport, here are called vectors. But to specify, this is a biological vector since it carries its pathogen inside of its body. Then define pathogen is a microorganism. So a microorganism which causes a disease. So anything that can cause a disease is a pathogen. Then Roman number 2, name the malaria causing pathogen. So the pathogen for malaria is called Plasmodium. So for this video, we are going to end here, but we are going to continue with more of these random questions. So stay tuned to this channel, subscribe, share, and come for online classes. So that's it for this video. Thanks for watching.